right, I'll go ahead and do some introductions. Hello, everybody. We are WISC, Women in Science at Columbia. Thank you so much for being here on your Saturday and coming to listen to this talk. We're going to hear from Eliza Yeager. She's a second year in the Biological Sciences Department, and we're excited to hear about her research field. So Eliza, what got you into science as a career?
It's actually a little bit of what I'm going to talk about today, which is like human origins. I thought it was so cool. I wanted to know about how people evolved to be as like complex and capable of many things as we are. And that kind of drew me to neuro because like the human brain is so special and interesting. And then I realized I did not want to study people because it's hard and takes a lot of time. And it's actually kind of difficult to get at the questions that I was interested in. And so, going through a couple different labs, a couple different species. I've worked with rats and mice, and now I work with salamanders. And it's a really good evolutionary model to figure out what makes the brain special and how we evolved and like how we're this awesome and smart.
Very cool. And how did you decide on choosing this particular lab over other labs you rotated in?
So I tried out two labs, including the Tosca's lab, which is the one I'm in now. The first one worked with worms, so like C. elegans, and I got to do a little bit of worm behavior. So like interesting behaviors are what I'm interested in and like how the brain creates them. But in my opinion, the worms, they just weren't doing interesting enough stuff. And so I wanted to go a little bit further up in the hierarchy and work with vertebrates. And so salamanders turned out to be the right choice for me.
Awesome. Thank you so much. Everyone, please feel free to interrupt with questions as we go through the talk. But if you're too shy to interrupt, as always, put your question in the chat box and I'll keep an eye out for it and I'll do the interrupting and ask it to Eliza. Let's see, anything else I should mention? I think that's all. Yeah, you can take it away.
Okay, cool. Yeah, so like I mentioned, I got interested in science because I wanted to know more about the human brain and what makes us special. And I took a couple anthropology classes, and a lot of the things that people mention in terms of why we're special is human language. We're actually able to communicate really complex and abstract ideas, like things about our own brains. We can cook food actually, which is a very specialized skill, and it's allowed our brains to develop in such a complex way. And we can use tools. You can see examples of tool use across a lot of different animals, but the extent to which people use tools, like, I mean, we're all on computers right now talking about complex ideas. It's kind of a great example of how special the human brain is. And so with all this, it's easy to kind of think of the human brain as king. We've evolved to the point where we have all of these complex capabilities, but there are actually a lot of similarities between the human brain and other vertebrate brains. So if you're a vertebrate, all that really means is that you have a spinal cord and a central nervous system to some degree. And so all of these brains, if you look at the different colors, actually have really similar parts. So starting with just, a fish or a frog, an alligator, a pigeon, rabbit, all of this pink stuff is what we call the cortex or the telencephalon. And it actually has a lot of similarities, even up to the amphibian point. So, maybe you've heard of people saying like, oh, the reptilian brain, it's so primitive. It's, everyone has a reptilian brain that guides their base instincts. That's kind of a weird way of thinking because the reptilian brain and the amphibian brain actually have a lot of similarities with the human brain. And so how does this happen? How have we evolved? And maybe if you Google evolution, which I did yesterday to find some good graphics, you come up with something like this, where it's this really linear progression of like, cave people, and that's always men, cave people evolving up until, this businessman on his phone. And this linear picture of how evolution has happened is not true. This is not really how we think about evolution in the modern era. And so usually to kind of get at the nuance of how evolution has happened, you have to look at maps that at first glance seem really complicated, but in reality, this is actually a really good way of looking at how all of the different species on Earth are interrelated and how they've evolved over time. So if you start here, which is when the Earth was born, when, cells or bacteria first appeared in the ocean and then started to kind of diversify, we have all of these different animal types until we get to fish, which are, the first vertebrates that we've been able to identify. And this part of the tree is actually where I'm really interested in looking, because these are the creatures that have they're vertebrates, so they have brains and they have spinal cords. And so they actually share a lot of similarities. And the way you read this is actually all of the animals, all of these branches that have reached this point, they're all still on the planet. Except, if you look in here, there's this little black spot where the dinosaurs went extinct. But actually, if you look, dinosaurs weren't that long ago. They were vertebrates, and we know they were reptiles. And so by looking at, similar species like crocodiles and turtles, we can actually figure out a lot about how dinosaurs may have lived, which is why we have all of these cool pictures of dinosaurs, even though, of course, we've never seen them. But what I want to draw your attention to here is that there's this branch point around here where you have the first common ancestor of amphibians, reptiles, birds, and mammals. And this is really where things would get interesting for an evolutionary neuroscientist like me, because if we knew what that vertebrate brain looked like, we could figure out a lot about what makes vertebrate brains, what keeps them together, what's their commonality, like what are the basic building blocks of the vertebrate brain that have allowed, mammalian evolution and human evolution to kind of explode and give us all these cool and complex abilities. And so ideally, we would go back in time and use a time machine and figure out, what was this animal? Who was this common ancestor? And what did their brain look like? Why were they so special? And that, maybe it looks something like this. There are a lot of artist renditions of like what these animals may have looked like. And, it was probably something like a fish with a spinal cord and, maybe some little nubs where it would have 4 limbs. But since we don't have a time machine, we'll have to make do with this comparative approach, which is what a lot of neuroanatomists do and what, evolutionary scientists of all different flavors do. And so here's our common vertebrate ancestor with a spinal cord in his brain. And what you see at this branch point, we could look at fish, but they don't actually have 4 limbs and they don't walk on land. So these are terrestrial vertebrates that I'm interested in. And so at this branch point, one of the most further back animals are these salamanders. And this is actually the species that I study, the Pleurodellus wattle newt. And, as you go a little bit further forward in time, you have, lizards and turtles, birds, and of course, mammals. But by looking at this furthest back species that of course is still alive and is no less evolved than we are, it's been evolving for exactly the same amount of time. But since it branched further back, looking at this salamander brain can actually give us a lot of hints about what this guy's brain may have looked like. So this is what we call the comparative approach in neuroscience and in evolution. Okay, so that's all well and good, but how do you actually compare these things? There are actually a bunch of different levels that we can compare different species at. So first you have the genome level. So of course, the blueprint of all life, each species has its own specific genome. And that genome determines how the brain, which is made up of neurons or these different cell types, how those cell types become the way they are, how they work the way they do. And of course, cells don't just, they don't exist in a vacuum on their own. They wire into circuits. And of course, all of these things are interrelated. So genes determine cell types and cell types determine circuits. And then if you finally get to the structures of the brain, so this is actually a mouse brain slice, you can then compare across different structures, what do they look like? Are they similar? Are they different? And then at the very final level, you can look at the behaviors that the brain is actually able to give rise to. So, in the mouse, here's like the hippocampus, which we pretty much know is responsible for a lot of spatial memory, a lot of learning and memory. And so you can go into a salamander and say, okay, is there anything in the salamander brain that acts like the hippocampus on a behavioral level? And then you can go back through all of these different things and compare the different levels. And so, like I said, none of these things exist in a vacuum. But the way that evolution works is, it can't directly act on your genome. It's not going in and making changes and being able to select for those changes at the genome level. Actually, the only place that natural selection can work is on behaviors. And so when I say behaviors, what does that actually mean? So things that natural selection could act on would be maybe your ability to find a mate and go and have more babies and have those babies then reproduce as well, which is what we call fitness. But fitness isn't just mating. It could be, feeding behaviors, your ability to navigate an environment, to find shelter, to hide from a predator. And all of these things are determined by how well your brain is able to work with the environment and figure out what your next move is, basically. And so the way that natural selection actually is able to select for things is if you have mutations in your genome. So say you have a little bit of a change in your genome that makes your neurons better able to find a mate or find food, then that's actually how evolution happens. Of course, this is all very difficult to actually pinpoint when you're looking at a brain. We can't actually, again, go back in time and see all of the different changes that animals have gone through. But what we can look at is the structure of the different brains. So this is a basic rendition of what this orange spot is in a mouse and a turtle. And what's important to know is that mammals, so mice and people, we all have these 6 layers of neurons. And 6 layers is a lot of layers. And basically, all of this different layering means that you can have a lot more complex computational power in your brain. It's just a lot of different ways for neurons to wire together and encode different information. Now, the turtle brain has 3 layers, which means that they're maybe slightly less complex in terms of the ways that the neurons can wire together. But if you think about a mouse and a turtle, both of them need to go and find food. They both need to find mates. And they're both capable of learning about their environments in a meaningful way. And so what this looks like in a bit more complicated level, so right now we're looking at these different cell types. So all of these different diagrams are basically showing what the layers actually look like in terms of how neurons wire together. And so at a basic level, there are 2 different flavors of neurons. There are neurons that can fire and they're excitatory, they activate things. And then there are neurons that are inhibitory and that can kind of keep the other neurons in check from firing too much and encoding too much information. And this is common across turtles, which is what this is representing. So turtles have these inhibitory interneurons that keep all the other neurons in check. And they actually have very similar types to a mammal or to a human being. And this is really cool because if turtles and mammals, or turtles and people or turtles and mice have similar neuron types, that's something fundamental about the vertebrate brain that's been consistently kept in check for hundreds of millions of years. And so this is a diagram showing basically how crazy and complex the primate brain can get, where you have all of these different synapses, all of these different layers trying to wire together to encode information. And this is, of course, the benefit of looking at animals that have less complex brains, because this one is a lot more simple, where you have all of this information and it's going straight into the hippocampus analog of a turtle. And so by being able to kind of prune back all of these complex synapses and just look at a more simple circuit model, we can actually figure out more what information gets to the hippocampus and how, if you look at a more simple model. And so this is how we get to salamanders. Salamanders actually don't have any layering as far as we can tell. So they have this one layer of those excitatory neurons in something that does a similar function as these other orange pieces in reptiles and mammals. But because they're even more simple than this 3-layered thing, we're even more capable of figuring out how all of the neurons work together to do the same tasks that this mouse model is doing. And so this is what an actual salamander slice looks like. So this is a slice of a salamander brain. This is the whole hemisphere. So if you think about the way that a brain is structured, it has those 2 halves. And if you slice down from the top, like if you were wearing a crown, this is what that slice looks like. And so it's actually pretty small. And these green bits that you see, those are neurons. And the reason that these neurons are green and then these aren't is actually changes in the way that the genes are expressed in these neurons. And so if you just look at the structure of the brain without any kind of gene information, it looks really similar. Everything's kind of just neurons. But if you go in and look at that gene level, like that most basic level that we were talking about before, you can actually find these differences and then kind of try to figure out, what is this kind of neuron, what's it doing, and how is it encoding information? And so that's what I'm interested in. And so just to, kind of take a step back and think about the kinds of things that brains really need to do. Each of these animal species would probably be capable of going through this maze, right? And using these cues, so like maybe it can see a flower or it can smell something that tells it that this is the right place to turn and navigate its way to some sort of reward at the end. Each of these species is capable of doing that. And so if we look at the salamander, if it learns this task, can get through the maze, and we look at how its brain works, maybe we can learn a little bit about how the human brain might do that. So if we zoom in here a little bit and look at what neural activity is doing, this is actually something we can do with the new salamander species that we create. We can cause the neurons to light up when they're active. So this is basically just a way of seeing how neural activity, what's going on as the animal is actually behaving. And this is something we can do. This isn't in salamanders. This is just kind of an example of what this might look like if you were imaging in a salamander as it behaved. So each of these different lights, that's a neuron. That's a neuron that's lighting up as it becomes active. And so you can do these recordings while the animal is behaving, and then go in and figure out which of these neurons were active and what the pattern might mean in terms of how it's actually letting the animal do the cool things that it does. And so if we can go in and figure out what that one layer, that simple layer of salamander neurons is doing, and maybe figure out a simple model of how the neurons and all the different neuron types wire together and how that actually occurs while the animal is behaving, then we have this fundamental concept of how vertebrate brains encode information and how this may have been conserved over evolutionary time, from that first common vertebrate ancestor all the way up until people. And so this can not only give us cool information about how salamanders work, but it can also tell us more about how the human brain works and maybe the very fundamental building blocks of how brains have evolved and how they continue to work, which can give us a lot of new insights into neuroscience generally. So that's why I love it. And of course, this is all from a tiny little brain that is nowhere near the complexity of a human brain. But because of these similarities at all of these different levels that we can look at, we can actually figure out a lot about the human brain from our salamander. So that is what I have.
Ooh, thank you so much.
Yeah.
I don't see any questions in the chat, but I have a question. So
Yep.
when you're looking at the lighting up readout for activation of the neuron, you also are wanting to map that to the schematic of the different layers.
Yeah.
How hard is it to tell which one is which? Which of those lighting up is an excitatory one? Which one is actually an inhibitory interneuron?
That's a great question. So you can do it by only putting in the lighting up gene in specific kinds of neurons that you determine. So that's one way that's actually really hard, because then you have to make the salamander species that only puts the fluorescent reporter in those neurons. But you can also do it by having all of the neurons fluoresce and then going in and taking up the brain and then staining basically 4 different neuron types in that slice, like the one that I showed. So you can kind of go in and look at it after the fact and say, okay, that one was super active. What's that one's identity? And you can figure out a little bit more information about it.
Overlap the
exactly. Yeah.
Okay, very cool.
Yeah.
I also have another question. It might be really stupid. So I was reading about rats and mice at some point, and I heard that in those type of studies, they only use male animals to map the brain function. Is that true for salamanders also?
It is not. And I think...
Okay, that's good.
Yeah, no, that worries me too for a lot of those studies. I'm not sure why they do that. But,
I think it's because of the hormones. It's easier to control the...
Oh, that's right, because the females, they change over time. Yeah, that's a good point. yeah, no, we haven't done that so far.
That's good.
So how old are the salamanders once you start working with them and collecting data? Like, are they like fully adults, or do you like raise
Yeah, so yeah, that's something I didn't talk about at all. But these guys, they do go through metamorphosis. So they're born as eggs, and then they develop into larvae that can only live in water. So they have little gills. But then as they grow older, which is about around 3 months, the gills kind of retract and they grow all of the four limbs and they lose their swimming tail. And once they've gone through metamorphosis, we consider them adult. These guys are actually really cool because they can regenerate parts of their body, something I didn't talk about at all. But like if you make a little hole even in the salamander brain, it can grow back. So this is likely because they're kind of always developing. There's no stage that we consider like, oh, this is true adulthood. They're always growing and they're always developing, which is one of the reasons we think that they can regenerate parts of their body. But yeah, once they've gone through metamorphosis, we can start doing these, adult brain studies.
That's so cool. So sorry, is that applicable for pretty much their entire body? Because I've heard that about their tails and their legs, but that's all I knew.
Yeah, no, salamanders are really like there's a bunch of different species of salamander that can do this. Not all of them can regenerate their brain. But yeah, actually someone in our lab is looking at how the brain figures out how to regenerate. And I mean, you can't take out the whole brain, like salamander will grow a new brain back, but you can take out parts. And yeah, it takes maybe like 3 or 4 months. And then as far as we can tell, the newly grown brain looks exactly like the brain it was before.
That is crazy.
And there's all sorts of stuff we're thinking about doing with that too. Like if a salamander learns something and you knock out that part of the brain, does it grow back the memory? Do the neurons know how to wire up the way they were before, or is there some like baseline information that they encode? We don't know.
That's so fascinating.
Yeah.
Does anyone have questions in the chat? You guys feel free to ask questions.
Yeah. They're pretty cool guys.
Yeah, otherwise I'll just keep asking questions.
That's okay too.
I'll ask a question. So I know that there are parts of the brains that are like very primitive that we share, like humans share with animals. And then there are parts that are uniquely human. So where in that range do salamanders lie?
That's a great question. And I might argue with the parts that are uniquely human. I mean, there are larger structures. So like one of the things that people point at is like the neocortex in people, or like the prefrontal, which is like all the bumpy brain bits like right at the front. And I mean, I don't know enough about human neuroscience, honestly, to say, but there are parts of the cortex generally that we see reflected in salamanders and in turtles. And it's kind of hard to put things on a spectrum or a continuum of like complexity or intelligence, because each species kind of has their own specialization. So I know one of the labs at the Zuckerman Institute, Dmitry Aronoff, actually just came out with a paper that shows that birds, or the birds that put seeds places and can go back and find them, they actually have very, very good spatial memory. So they have a very highly developed what we might call, and I might get flack for this, a bird hippocampus. So that there are different things that birds and maybe rats or people are good at. And I guess what you might say people are good at is like the complex abstract thought, which, yeah, that is pretty unique.
Very cool. So what kind of behavioral tasks do you test them with?
So I'm still trying to figure out what they like and what they don't like and what motivates them. So I've tried to tempt them with food. They don't seem to care. But I'm trying. One of my goals, yeah, they like if you stop giving them food, actually they're really good at adjusting. They have a very strong metabolic capacity. But one of my goals is to be able to do like the kind of the maze idea where you give them different cues and ask them to find a reward or like a goal and then just measure neural activity as they do that. But that's still like in the works. People haven't used these guys too much for more than like neuroanatomy or developmental studies. So getting them to do behaviors is kind of, it's hard and it's not something people have really tried before. So I'm still figuring it out.
Very cool.
Is there a reason for that? Like, it seems like it's such an interesting species. Like, I'm surprised it hasn't been studied more thoroughly with behavior.
Yeah, I mean, people go and like watch them in the wild. Cause so these guys they live in like the Iberian Peninsula, like Spain. And you know, people have known about the regeneration thing for a while, but the reason we can actually do neuroscience with them now is because we can sequence their genome and actually create like the transgenic salamanders, like the ones that have like the fluorescent indicators in their neurons. So I think, you know, as we get all these cool new tools like CRISPR and different ways of editing animals, we are going to see a whole host of new, you know, animal models that we can use for neuroscience research. So it's not all about mice and rats anymore.
It's crazy to think of like trying to figure out what a salamander would use for motivation. Like that wouldn't have ever occurred to me. Like, okay, do we do food? That's so strange.
It's a huge part of what I'm trying to figure out now, because I was so sure the food was gonna work. Everything was gonna be fine. They were gonna love it, and they're
like, no.
Didn't care. Yeah.
What about language? Do they communicate?
I don't. They have some vocalization. Like, well, you know, they have. I don't think they have mating calls, but they can make sounds like if they're upset, or I think it's more just kind of like an alarm call. But they communicate with, you know, pheromone like odor signals, so they can recognize each other and recognize like that's a male, that's a female. And they have a very complex mating dance that they do where they twirl around each other. It's, yeah. When I watch them in the tank, they're always kind of moving around and exploring. So I think they're actually quite curious.
Are you able to house them together, or are they territorial? They're not territorial?
They're not. I mean, they're called the Spanish ribbed newt because they actually have poisonous ribs that can poke out if they're.
Sorry, what?
Yeah. They have a prey response.
My new favorite species.
I know.
Poisonous ribs. That's awesome.
I've never been ribbed before, but I'm always in fear. Yeah. Wait, I forget why I'm talking about the ribs.
Oh, are they territorial or
not? Seen. They fight when they're babies. So actually, this was something because we're trying to figure out how to breed them and like maintain our own colony. They will eat each other.
Oh no!
But not because they're, you know, angry or territorial. Mostly it's just because they're hungry and don't really have very good kin recognition when they're younger. But on the plus side, if they don't eat them entirely and they just eat a leg, the leg grows back.
Oh my gosh.
The evolutionary, you know, drive for the regeneration.
Yeah. Actually, one of our guys had one of his front arms bitten off, and then it grew back two.
Oh my god.
Like bifurcated
2 arms and one on one stalk.
This is crazy.
So
he's
cool.
still alive. He's doing great.
Wow.
So if you said they have these poisonous, I forget the word that you used,
Ribs.
But if ribs, yeah. So if that like pierced you or whatever, I'm assuming that's not dangerous to humans because they're probably meant for like much smaller things, or.
You know, I don't know. No one's ever told me that we need like an antidote. So I'm.
Yeah, it's probably, I guess,
probably
similar
fine.
to like a lot of
like,
other things where it's meant to kill like much smaller organisms,
yeah.
I guess. Yeah,
I think so. Because these guys get eaten, especially when they're smaller. They can get eaten by like fish or even other salamander species.
Oh, interesting.
if you've been to the zoo and you see like the enormous hellbenders, they're literally legitimately like this long. Yeah, they can eat them too.
Yeah, that is smart, because I mean, as an animal structure, they're a little bit soft and vulnerable.
Yeah. They don't have a skeleton. They're all cartilage.
Oh shoot.
Yeah.
Wow. So maybe that's another question I have. Like, what's its closest relative that has actual bone as part of its spinal cord? I mean, that's kind of a very specific question, but like.
No. I mean, it's probably reptile, like the turtles, or like the bearded dragon guys. Because I mean, these guys live most of their, like, at least the way we raise them, they live most of their life in water. But they are capable of going and walking around on land, but they can't be too far from water because they dry out. But that's another thing that we're really interested in, is like this transition between water going animals and land going animals, and what kind of changes in the brain actually let you figure out how to navigate in water versus on land. And like what that transition was in the evolutionary timeline.
Yeah, or the learning how to walk versus swim. That'd be really interesting to see, like different regions of the brain.
Well, and like when I take my guys out of water for the first time, they get kind of confused, but they they know how to walk on land. It's something that they just innately understand, but they're a little awkward at first. But yeah, definitely like that difference is something that I'm interested in too.
Cool.
Very cool.
Are there any more questions from the audience? Okay. I guess if there's no more questions, we can wrap up. Yeah, thank you all for coming. If anyone wants to like rewatch this, I think Aubriana, it'll be on YouTube, right? And
channel, it's just called STEM Starters.
Yeah, and then just keep an eye out for more emails. We'll be doing another one in a few weeks.
Yeah, thank you so much, Eliza.
Yeah,
Thanks,
of course.
guys.
Thanks, guys.
Thank you, Eliza. That was really cool.